Problems Australia has is every 4 or 5 years there's a mouse plague. We have lots and lots of rabbits in Australia, and all of which influence agricultural output. We usually use biocontrol agents to control the rabbit population, such as myxoma, calicivirus, but both the rabbits and the viruses are co-evolving to live happily together without major effects. So the idea was we needed a new biocontrol agent. The idea was to make a virus that only infects rabbits or only infects mouse, that causes immunosterility. Our modeling systems say this would be a very, very useful way of controlling the population, and for some reason it would be humanely rather nice rather than killing them. So what the idea was to make a virus that encodes a gene for a protein involved in reproduction. And the idea was the virus would infect the animal would make antibodies to the encoded protein and cause infertility. It was a nice idea, and we went about it, and the protein that we used was called ZP3, and it's found on the ovum. And it's the docking point of the sperm in this case. The idea is the animals would make an antibody like we would make an antibody to influenza vaccine and prevent fertility. In fact, it worked remarkably well, surprised us all. Those animals that were infected with a mousepox virus, which contained this ZP3, were infertile for the lifetime of the animal under those circumstances. The rabbits had the same effect under these circumstances. So we had developed a new technology which caused infertility. It's the next step that we added to this that caused all the controversy. And I'll go into that now, but there's a lot of bright individuals in this audience, and I wonder if some of you have already picked up a dual-use issue that we missed at the time. And that dual-use issue is we've created a transmissible virus that causes infertility. That was never discussed at the time. And that's fascinating. In other words, I think they actually made a science fiction movie, Men of Boys or something like this, where the world was made infertile by a passaging virus. So here's an example of dual use that we ourselves have missed. We published without ever recognizing it. What we did recognize, however, was when we tried to increase the potency of these viruses that caused this immunosterility. And we added another gene called interleukin-4. And this gene is a cytokine. It's used by the immune system to stimulate antibody responses. So the theory was that we'd get better antibody responses with these virus with this gene in. In fact, what we did find was not that the result, but the result was the viruses were highly pathogenic, and it killed mice rapidly, and even mice who were genetically resistant were killed with this interleukin-4 virus. The interleukin-4 itself suppressed the immune response, and that's why it was killing the virus under these circumstances. So basically what we had shown is that we had a greatly increased pathogenic virus that antiviral drugs didn't work against. We had showed that later. But more importantly, and this was the staggering effect that shook us at the time, was that vaccinated mice that would normally resist an infection were not resistant to this interleukin-4 virus. The interleukin-4 virus killed the vaccinated mice. And it was at that point that the researchers identified what is now known as a dual-use dilemma. The dual-use dilemma in biological sciences was not recognized in early 2000, but we said there was a problem. Now, how did we deal with it? Well, basically, we asked all our colleagues, what should we do? Shall we publish, or shall we keep it secret? And without going into too much detail, in an audience this size, it was presented to all our academics. And the general consensus, the majority, almost 100% consensus, was to publish. So we did ask our colleagues, and the answer was, there's enough out there that you should publish and let everyone know what is possible under these circumstances. In fact, the great Frank Fenner, who was involved in the eradication of smallpox, also had this view. So what happened next in terms of history? We submitted the article to the Journal of Virology, and it was accepted for publication. Around a little time before the publication, there was a roving reporter from New Scientist called Rachel Novak came around and wanted to know what interesting things we were doing in research. And I told her all about our great work in prime boost immunization with HIV, and she didn't seem that interested. But I said, oh, we've got this paper coming out that shows that vaccinated mice can't be protected against this virus. And her ears picked up like no one's business. This was an article that she could get her teeth into. And she wanted to write about it. We obviously couldn't say no. We accepted that because it was coming out in press in a month's time. And she wanted to write the article in the New Scientist. We asked if the article could be sent to the institutions concerned a week beforehand, so that we could look at it. I didn't think it was too dramatic. It was dramatic in terms of New Scientist, but the institutions themselves said in their bureaucracy, it's a common feature of bureaucracy to try to protect themselves, we better advertise it ourselves. We can't wait for New Scientist to come out. We will announce these results ourselves to the press. And when those results were announced, I think it went viral around the world under those circumstances, that this was the example of what biotechnology can do under these circumstances, and this was a major, major concern. Now, I don't know if the institutions themselves created the problem themselves. It's not an experiment you can redo, but it may not have made the headlines it did had the institutions had just accepted the New Scientist. But that would be a guess under these circumstances from that point of view. The rest is history. In other words, the paper is quoted as a dual-use example under these circumstances, but what I haven't talked to people about is some of the experiments that we did after that, after the papers. And those are the experiments we chose not to publish. In other words, we did make an individual choice not to publish certain papers. And I can just briefly touch upon these from that point of view. Many people thought that the virus was so pathogenic it wouldn't transmit. In fact, it does transmit. Transmission of this virus occurs exactly 7 days after infection. And if the mouse lives that long, it will transmit. So it does transmit. One of the very interesting findings that we made, and I won't go into the details why it's interesting in terms of biosecurity or elsewhere, was when we looked at these animals, these vaccinated animals, being vaccinated, infected with the IL-4 virus, when we looked for virus in these animals, and these animals died about a day later than normal non-vaccinated animals, so they're killed very quickly. When we looked for virus in these animals, we couldn't find any. Vaccinated animals challenged with the IL-4 virus lacked, or seemed to lack, any virus in their system. That was a remarkable finding, and we looked very, very closely for this. And we could detect very, very low levels of virus. 100, 1,000 plaque-forming units of virus was present in those animals, and that's quite remarkable. So these vaccinated animals actually aren't dying of the virus, they're dying of the toxicity of the interleukin-4. And that's really quite a significant finding from this point of view. We also looked at these vaccinated animals, although have little virus, they still transmit. We aggressively, and I really mean aggressively, vaccinated these animals to see if we could find a vaccine that would protect against interleukin-4. And our most aggressive vaccine protected about 50% of the animals in transmission. But interestingly, the other 50% of animals actually developed a persistent infection. They didn't die. It was though there was a battle between the virus and the immune system, and they lived their life with a permanent pox virus infection. Now,